So our next speaker will be Anton Lavrinienko from ETH Zurich in Switzerland, with the importance of metadata standards for integrative microbiome research. So please, Anton, go ahead, and thank you for being here.
Thank you very much. Can you hear me?
Yes.
Can you see the slides?
Yes. All
right, perfect. Then we're good to go. Okay. Hello, everyone. I'm Anton Lavrinienko. I'm an ETH fellow and a postdoctoral researcher in Nicholas Backlish's lab at ETH Zurich in Switzerland, where I also work on the microbiota Vault launch. And thank you very much for your invitation. And it's my great pleasure to be here today and to see so many inspiring talks. The microbiota Vault is in the launch phase, and this comes with a number of challenges, but also opportunities to develop various standards and guidelines that will shape the future of this global initiative. And thanks to Pascal's talk, we now have a really nice overview of this effort, which makes things much easier for me. In my talk today, though, I would like to draw your attention to one specific aspect related to the contextual information associated with biological samples, also known as sample metadata, that Pascal briefly mentioned in her talk. And I would like to emphasize and highlight some important aspects of the metadata for microbiome research. So it's unlikely that I need to convince anyone here today is that the microbial communities are, especially those residing in the human gut, are important for health. But we now know quite a lot about factors that contribute to the loss of microbial diversity, and the microbiome appears to be particularly vulnerable to industrialization. And when comparing samples that originate from two distinct populations, for example, like you see here in a slide, the pristine population and disturbed population, or population that levels differ in the level of industrialization, a typical microbiome analysis will end up with something like this. So a massive amount of not so human readable data, which is not that exciting overall, really. But with addition of the sample metadata, with all the valuable contextual information, such as the who, the what, when, where, and why about these samples, it becomes possible to draw meaningful conclusions from comparing sequencing data that originates, for instance, as in this example, from industrial and traditional human populations. So when we analyze the microbiome data of any kind, of any source, from any sample type, our task is to really put the observed microbiome community back to its environment and account for diverse phenotypes, the experiences, the exposures. All this can be achieved with the use of high-quality metadata. Of course, the type of the metadata, though, collected in various studies is highly dependent on a sample type. For example, in human studies, it can be important to account for differences in diet, geography, lifestyle, host age, various confounding health conditions, medication use, and so on. In repeated observations from human populations, or for instance, in wildlife, seasonal changes can be also important to account for. Whereas studies in laboratory animal models, like in mice or in rats, they should consider also animal origin, like a vendor, and co-housing strategies, cage effects, and etc. So what is really important is that the metadata, where we collect the metadata, it should be standardized. So in most studies, the metadata is structured as a matrix shown here, probably familiar to everyone, like a simple spreadsheet, where each row is the sample metadata and each column is a property of the sample, such as body site sample, or host age, or host sex, treatment group, and so on. So on the first glance, this looks good. The things are looking strong here. But as researchers are frequently use free-form fields and often without any validation, the metadata is prone to errors and inconsistencies. And here they highlight with orange, we start to see different misspellings, incorrect missing data, and etc. Which at the minimum can be concerning and confusing, within a single data set, but then things go out of hand and further complicated by the differences in formatting or culture across data sets, across different disciplines and domains, institution, countries, and etc. And as such, lack of standardization can really hinder data reuse and highlighting for the need for wider adoption of metadata standards in microbiome research if we are to really capitalize on this huge volumes of incoming sequencing data. So the good news is that the metadata standards exist and a number of really tailored checklists that are suitable for microbiome research are out there. Perhaps one of the best examples that I would like to show here today is the family of standards developed by the large community effort led by the Genomic Standards Consortium, describing the minimum required information about genomic sequences, and they organize this information into a checklist, and each checklist capture information most relevant for a certain investigation type, be it marker gene sequencing or so-called amplicon sequencing or metagenomic sequencing or any other kind of sequencing. These checklists are later on can be used in combination with environment-specific packages or extensions to collect a more comprehensive range of environmental parameters which are tailored and can help to better contextualize and describe a given sample. So at the minute, this includes about 23 extensions with the most recent additions for food and agriculture. So everyone can really find something suitable for their data sets there. What is really important here is that these checklists and packages, they use control vocabulary to describe the host organism, environment, or sample processing strategies with the most appropriate terms. And many of the fields in the mixes are also relying on ontologies, making the metadata more logical, more searchable, more machine readable. So all these attributes that we want to actually see for data-driven science. All right, so based on the data from the SRA and ENA data archives, we can see the use of standards across research communities. And the adoption rates for these standards across the community are not great, but they're fairly high. What is interesting that it appears that the usage of the mixes packages that best describe the reported sample type by the original author, here they are marked in the proportional plots, they marked in red, expected mixes package is somewhat higher for environmental sample types in ENA, so in European Nucleotide Archive, compared to the SRA from NCBI in the US, which where the mixes packages are more frequently used for human-associated samples. So the use of other checklists here in blue, for example, the default ENA and NCBI packages is not necessarily wrong or bad, but oftentimes they're limited to very basic project descriptors and less comprehensive in their nature. They're less comprehensive in biology, if you will. So an NCBI by sample search for putative human microbiome records resulted in over 1.6 million records earlier this week. And the scale of microbiome data being shared by the community is rapidly growing, which is really great, which is great news, right? But if we really look into this more closely, there are bad news. The bad news is that there's still a number of significant challenges and barriers for data reuse due to a high level of missingness, absence of very basic information associated with this human microbiome samples. So in this plot here, a metadata mapping effort from the public archives revealed that patterns matching host age. So here on one axis, you can see a missingness score, so in percents, and on the other axis, you can see all kinds of expressions and terms which are mapped to age. So they may be a bit misleading if you read them, but don't bother about it. The main point is that anything which we can map, which is resembling age, is represented here. And what we can see is that the more than 80% of missing values are associated with age in the public repos, which is really, really shocking. And the similar trends observed in host sex and also sample type, for example, whether it's a case or control, or whether it's a sampling site, gut, or skin, and so on. So sometimes this probably happens due to the lack of incentive to report beyond the bare minimum information requested upon the data submission. intentional or unintentional underreporting, or reporting using non-standard terms, which are not mappable in the end. And even simple misunderstanding oftentimes, I believe, can be a reason for this. When, for example, a certain field has no variation within a data set that researchers put online, and some researchers may see no value in copy-pasting identical data across rows and rows and rows of samples as they are homogeneous, such as age, for example, or sex. So yet this information is really critical when comparing across data sets. But let's step back for a moment and really think, why don't we share our data? There are a number of distinct perceived barriers to sharing any scientific data, including metadata in microbiome research. And here they're presented by 12 distinct points, each with a separate icon. Few of these come up more often than others, and they include concerns when sharing sensitive data, fears of being scooped, and that the process is difficult and unclear. So together, all this kind of boils down to these three large themes: emerging groups, knowledge barriers, reuse concerns, and lack of incentive. So it ultimately creates a scenario where it is impossible to do anything without available metadata. Or it can be very difficult to interpret and reuse the heavily fragmented or incomplete data sets to reproduce original findings, not to even mention significant bottlenecks for data integration, comparisons across data sets, and the efficient reuse of microbiome data. So the fact that the data archives are half empty highlight the need for more stringent submission process that would encourage researchers to share more data, especially when it is not a matter of ethical privacy concerns. Some researchers call for the wider adoption of more comprehensive minimum standards rather than standards themselves, with such data being fair and resting kind of on a sweet spot here between comprehensiveness and feasibility, as if we ask too much and be overambitious, we may end up asking something not feasible from research community. Ultimately, the good practice would allow other scientists to reproduce their regional research at the minimum, and ideally also to facilitate research beyond the regional scope. So now, saying all this, I would like to kind of tie it and bring it back to the Microbiota Vault project. And the overarching goal of the Microbiota Vault launch phase is to optimize various steps of the workflow, including the development of minimal metadata requirements for sample submission. So right now, this includes 47 fields in the first version of the minimum metadata requirements for Microbiota Vault, with 21 metadata fields that are required for a compliant report. For example, this includes sample identifiers, environmental context information, spatio-temporal metadata about where the sample was collected and when, and technical notes to control for cold chain during shipping and different sample condition information. And an additional part of metadata is dedicated to sample provenance. So the depositors are clearly identified, and their affiliation and their persistent identifiers are clearly marked. We also offer 13 recommended and 13 optional fields, each to accommodate more comprehensive metadata where available and possible. And then when the scientists would be willing to share this data, as we explained to the sample depository in our user guide.
2 minutes, Anton, until
yes,
the time
thank you.
is.
They are not mandatory, these fields, they're recommended and optional fields. But they are definitely increase the sample value, as we explained to the sample depositors. So collecting and sharing the high quality metadata is clearly rewarding, and it has a number of advantages, as exemplified by Earth Microbiome Project, American Gut Project, Tara Oceans, DiBimmune, a number of other large-scale coordinated efforts. And what we hope with this to inspire for better metadata reporting practices within our community, and that learning from both past mistakes but also success stories when it comes to the metadata will help to secure this scalable and sustainable future for the Microbiota Vault initiative. Thank you.
Thank you very much, Anton.